Welcome to the Mechanisms of Developmental Patterning module. In this talk, we're going to look at a type of specification called as conditional specification. We will first look at the process of specification and the different types of specification, and then we will concentrate on conditional specification. The different stages that are involved that enable cells to determine their fate in the body include the stage of cell commitment. Now, cell commitment requires the process of specification to occur, followed by the process of determination. Once cell commitment has been finalized, then the cell undergoes differentiation, which allows the cell to develop specialized structure elements and distinct functional properties. Now let us look at each one of these stages. Specification is the first stage of commitment, where a cell is deciding what it's going to become in that multicellular organism. In order to understand specification, we have to go to the cells when they are in a blastula. In the blastula, the cells, which are the blastomeres, are not showing any characteristic properties of the cells that they will be developing to in the future. So if we take one cell from the blastula that we know is going to be a muscle cell further on in development, what we do is we take the cell and we put it in a neutral environment and look at what happens to that cell. If that cell becomes a muscle cell, then we say that that cell has undergone specification. Another example of that is, instead of using a cell that we know becomes a muscle cell, what if we took a cell from a blastula and when we culture it in a neutral environment, it becomes a neuron? Then in this scenario, that cell has undergone the process of specification because it has begun the process of becoming a neuron. Thus, in the case of specification, if a cell that is in a blastula is able to differentiate towards a given fate when it is in a neutral environment, then we say that specification has occurred. There are three main ways by which specification can occur. The first strategy is called autonomous specification. And in this scenario, the cell in the embryo has cytoplasmic determinants, which are usually in the form of mRNA and proteins that function to commit the cell towards a specific fate. Another type of specification is called conditional specification. And in this case, the cell in the embryo is able to commit to a specific fate based on cues that it gets from its environment. Thus, its fate depends on its position and interactions with the other neighboring cells and molecules that it comes in contact with. The third type of specification is called syncytial specification. And in this case, the cell fate is determined by the location of these cytoplasmic determinant factors like mRNA and proteins and the position of their nuclei in a structure called as a syncytium. The second type of specification is conditional specification. In this process, the cells achieve their respective fates by interacting with other cells. In the case of autonomous specification, the cell did not have to interact with other cells to determine its fate. However, in conditional specification, the cell's fate is determined based on its interaction with other cells. The fate is determined by various factors that include cell-to-cell contacts, secreted signals from cells like paracrine factors, and physical properties of the local environment like mechanical stress. An example of conditional specification is shown in the figure below in the blue box. Here in the top part, we have a blastula where you have numerous cells available. If we take cells that are shown that are going to be normal back cells and put those cells into a part of the blastula that is different from the origin? When we look at the embryo, we find that the cells are going to form whatever the neighboring cells were. So, if we look at the cells from where they were supposed to form back tissues, if they were transplanted to the part where a belly is supposed to form, those cells go ahead and form a belly because that is the cues that they are getting from their neighboring cells. On the other hand, if another experiment is performed where you remove a whole chunk of cells from the blastula using a glass needle, you would expect a larvae to have missing portions. However, the larvae develop perfectly fine, and in this case we're looking at the larvae of the Pluteus family. So in this case, even though we had missing blastomeres, it did not affect the development of the larvae because the cells were able to compensate for the loss of those other blastomeres due to the process of conditional specification. Let us look at some experiments that were performed in the past. The first experiment was performed by Roux, who wanted to demonstrate that the only type of specification that occurred was autonomous specification. The way he showed that was he destroyed one cell of the two-cell embryo and looked at the development of that damaged embryo and found that it developed only to form one half of the embryo. His experiments seem to indicate that different cytoplasmic determinants are separated with the first cleavage where the single-cell zygote forms the two-cell embryo. Looking at his experiments, another embryologist, Streich, decided to do another set of experiments. And what he did is he took the blastomeres present in the four-cell embryo and he looked to see whether they could develop into complete larvae. What he showed that all four cells from that blastomere were actually able to develop into larvae. And so the way that he could explain this finding was that a cell's fate can actually depend on its neighbors. That was the only way that intact larvae could be made from each single cell of the blastomere. It should be noted that the larvae did have some differences, but the differences were quite subtle. Another experiment that was done was to observe what happened to the larvae if the cell arrangement was changed in an embryo. This was done by subjecting the embryo under pressure between two glass plates so that the way cleavage happened was changed. What was found was that the embryos developed into normal larvae once again. Thus, the relative position of a blastomere in the embryo can determine its fate, and this is through cell interactions. Thus, these cell interactions were critical for normal development, and thus, scientists and embryologists were able to prove that conditional specification is another strategy for the process of specification.
With this, we come to the end of our talk where we learned about the different types of specification and what happens in conditional specification.